Hello, students, and welcome to another class live TA session for practice problem set week 9 in the course Basics of Biology. So it's already 3 p.m., but since the population is thin, we'll wait for 5 more minutes and I'll keep admitting students who are trying to request to join this link. Then we will start the class at 3:05. Thank you. Okay, students, it's already past 3:05. We will start the class now. I welcome you all to again another TS session of Basics in Biology, where we'll be solving practice problem set for lecture set of week 9. So we'll be dealing with week line 9 lecture sets for circulatory system and the muscular system. So today the questions will be about circulatory system and the muscular system, and these will resemble your assignment questions, the quiz questions, and the kind of multiple choice questions that you are going to face in your exams. Okay, so without any further delay, let me first introduce myself. My name is Ashutosh Behera, and I'm a PhD student PMRF scholar at IIT Kanpur. This course is taught by Professor Vishal Trivedi, and he's at IIT Guwahati. So these are recorded lectures, and me, along with other PMRF scholars, are the teaching assistant for this course. So this is the pattern that we follow up until now, and this is the course of my class. That this is a problem solving session, and this will be an interactive session. But I would request all the participants who have joined the live class, as well as students who will be watching this video, to turn off their video and mic and communicate with me via the chat box. So what I'll do is I'll display my presentation. I will display all the questions relevant to this session, multiple choice questions, and I will ask students to guess what is the correct answer, to tell me what is the correct answer. Then I'll reveal what is the correct answer, actual correct answer to the question, and we will understand briefly about the concept behind the correct answer. In that way, clearing the doubts of covering all topics and clearing the doubts of all students. If you have any query, any doubt, or you want me to repeat any question, feel free to stop me in between and keep communicating with me via the chat box. Okay, now let me share my screen with you, share my slides so that we can start the class. Just give me a minute. So I hope everyone can see my slides. The students present in live class. I think there is only one student with us till now. Can you see my slides? Can you see the slides moving? And is my voice loud and clear? Loud and clear and audible? Let me know in the chat box with a yes or no. Then we will proceed with the question and answers. Okay. Thanks for the heads up. So once again, I welcome you all to week 9 for Basics of Biology, NOC 25BT02. The instructor for this course is Professor Vishal Trivedi, and my name is Ashutosh Behera. I am the teaching assistant for this course. I am a PMRF scholar at IIT Kanpur, and this course is brought to you by NPTEL. So what we'll be solving? We will be solving multiple choice questions, exemplar problem questions from lecture set 9, from lecture set of week 9, where Professor Vishal Trivedi has extensively talked about the circulatory system and the muscular system. Okay. So we will follow the same trajectory that I explained before starting the class. I would request all the participants to please turn off your mic and video chat and communicate with me only through the comment box. Let us keep this session interactive as well as smooth conducting of this session. Please keep interacting with me answer the questions that I'll be posting, and engage in discussion. Ask me your doubts, feel free. This is the only point of live interaction with the NPTEL course that the NPTEL has done. Okay, without further delay, let us look at the first question of week 9. The first question of week 9 is, which of the following have an open circulatory system? This is the first question. Which of the following have an open circulatory system? Option A, arthropods. Mind the spelling mistake. Option A, it's arthropods. Option B, porifera. Option C, nematoda. And option D, annelida. These are the four options, and the question is, which of the following have an open circulatory system? To all the students who have joined recently, I would request please turn off your video and mic and communicate with me through the chat box. Post your answers in the chat box, and I will be following the comment box routinely. Which of the following have an open circulatory system? So what we are dealing with is lecture week 9 for circulatory system and muscular system lectures. We have 15 set of exemplar questions similar to what you will face in your quizzes and assignments or your tests. These are some of similar questions from the same lecture series. This will help you clear your concepts as well as familiarize with you with the style of questions that you will be getting in your quizzes and assignments. Okay. So the first question is, which of the following have an open circulatory system? And the options are A, arthropods, B, porifera, C, nematoda, D, annelida. Let me know what you think is the correct answer to this question in the chat box. I will wait for a minute for all those who are attending the live session as well as those who will be watching this recorded class. Let us wait for a minute and collect some answers. I will reveal what is the correct answer in a minute or so. I would encourage everyone to think of the answer and participate in this class. Okay, it's time for me to reveal the correct answer. Which of the following have an open circulatory system? The correct answer to this question is option A, arthropods. Option A, arthropods is the correct answer to this question. Arthropods have an open circulatory system. Now let us understand two major kinds of circulatory system. One is open circulatory system and a closed circulatory system. What is the difference between them? In an open circulatory system, the blood is pumped into what is known as hemocoel, with the blood diffusing back to the circulatory system between the cells. So everything is open, blood is pumped into the body fluid, which is the hemocoel, and the blood diffuses back into the circulatory system between the cells. So you have such a structure which is pumping the blood, and then it is directly bathed into the cells, and then the cells blood diffuses back to and fro from the cells. The open circulatory system is common to mollusks and arthropods. So two species or two kingdom of classification, two phyla of the animal kingdom show the presence of an open circulatory system, mollusks and arthropods. Closed circulatory system, mostly vertebrates and few invertebrates have a closed circulatory system. In closed circulatory system, blood is pumped by a heart through vessels and does not normally fill body cavities. So the blood does not fill the body cavities inside the closed circulatory system. Whereas in the open circulatory system, blood is everywhere in the body cavity. So in the closed circulatory system, we have a heart, we have arteries supplying blood from heart to the body cells, and valves and veins supplying blood from the cells to the heart. So the open circulatory system is present most commonly in mollusks and arthropods, while closed circulatory system is present in vertebrates and a few invertebrates. So these are some of the features, key features of open versus closed circulatory system. The blood flow is in open spaces called sinuses, whereas in closed circulatory system, blood flow is confined in blood vessels only. What is the efficiency of open circulatory system? It is very less efficient. Efficient in the term of gaseous exchange. Closed circulatory system is more efficient. Organisms, which organisms have open circulatory system? Insects and mollusks. In closed circulatory system, humans, fish, birds, all the vertebrates have closed circulatory system. Blood pressure. What is the blood pressure within an open circulatory system? Obviously it is low, while the blood flowing inside arteries and veins have a higher pressure. Oxygen transport, as we just discussed about efficiency, oxygen transport is slower in an open circulatory system, while it is faster in a closed circulatory system. So arthropods, example, insects, crustaceans and spiders, all these belong to the class arthropods, have an open circulatory system where blood or the hemolymph flows freely in body cavities instead of being confined in the blood vessels. So this is what the crux of open circulatory system is. Porifera, what are the other options? There are other options were Porifera, Nematoda and Annelida. Porifera or sea sponges does not have a circulatory system. They rely on water flow for nutrient and gas exchange. In a porifera or in a sponge, the water flow itself acts as a pseudo circulatory system which allows for nutrient and gas exchange. In nematodes or roundworms, they have simple unsegmented body and lack a circulatory system. They also use diffusion for transport. And in annelids like earthworm and leech have a closed circulatory system with blood confined to vessels. So annelid is higher organism which has a closed circulatory system. Nematode lacks a circulatory system. Even porifera does not have a circulatory system. So the correct answer is arthropods which have open circulatory system. Now is it clear to the students? Is it clear to the students? Just let me know in the chat box with a like or a comment or a yes. If the concept is clear, then we will move on to the next question. Okay, okay, we will move on to the next question. Question number 2. The simplest vertebrate heart is a dashed. The simplest vertebrate heart is a option A, one chambered heart. Option B, two chambered heart. Option C, three chambered heart. Or option D, four chambered heart. How many chambers does the simplest vertebrate heart have? Is it one chambered? Is it two chambered? Is it three chambered or four chambered? I think this is a memory based question, but if you remember the phylogeny of simplest vertebrate, the phylogeny of heart, evolution of heart, you would know this answer. How many chambers does a simplest vertebrate heart have? Let us wait for a minute or so and collect some answers. I will reveal the correct answer in a minute. Okay, I think it's enough time and time for me to reveal what is the correct answer to this question. The simplest vertebrate heart is a option B, two chambered heart. The simplest vertebrate heart is a two chambered heart, and two chambered heart belongs to simplest vertebrate, which is fishes. Fishes have two chambered heart. Now let us see the phylogenetic tree of a vertebrate uh, vertebrate phyla with a comparative anatomy of vertebrate hearts. These are the circulatory circuits for all the above mentioned vertebrates. The most primitive or simplest vertebrate has a two chambered heart, which is a fish. Fish has one circuit, a single circuit, two chambered heart. Single circuit means there is only one circuit of blood of circulation from gills to the body and body to the heart. and then heart to the gills again, connected in one circuit. Then comes in evolution two circulatory loops, three chambered heart. Then comes the three chambered heart with two atriums and one ventricle. This is the heart of a frog. It has two circuits, one to the lung and back, and then another circuit operating from the ventricle to the body and back to the heart. So the heart becomes the junction of the circuit. Two circuits, three chambered heart. Then farther in evolution we have turtles and lizards, which are reptiles. Lizards are reptiles and turtles, amphibians. Two circuits, five chambered heart. Ventricle divided into chambers. Now the single ventricle present in the frogs is now divided into five chambered heart. And it still has two circuits, one to the lung and to the back, and from separate compartments pumping the blood back to the body and getting waste from the cells, deoxygenated blood from the cells, and putting it back inside the heart. Then comes higher reptiles like crocodiles, which have two circuit, four chambered heart, two atriums, two ventricles, and two circuits. Then comes birds, which have two circuits, four chambered heart, and mammals, which are two circuits, four chambered heart again, two atrium, two ventricles, one circuit completing the lung circuit, ventricle to the lung and then back to the atrium, while the other completing the body circuit, ventricle to the body and back to the atrium. So this is the comparative anatomy of all vertebrate hearts, and the simplest vertebrate fish has one circuit, two chambered heart. Now looking at the two chambered fish heart, this is the circulatory system of a fish. The fish have gills here. The red depicts oxygenated blood and the blue is the deoxygenated blood. Oxygenated blood moving through systemic capillaries, getting pumped into the atrium, heart chamber that received blood returning to the heart. So atrium is the chamber which is receiving the blood that is returning to the fish heart. And then we have another pocket or another chamber in the heart, which is the ventricle, heart chamber that pumps blood out of the heart. So two chambered heart of a fish, atrium collecting the blood returning from the cells to the heart, and then ventricle pumping the blood out of the heart. So this is the simplest circuit, one circuit, two chambered heart of a simple vertebrate fish. I hope it is clear to the students. If yes, just let me know with the chat box. With the yes or no in the chat box. Okay, good. Moving on to the third question of this TA session. How long do RBCs, red blood cells, live in a healthy individual? How long do RBCs, red blood cells, live in a healthy individual? Option A, 20 days. Option B, 120 days. Option C, 180 days. And option D, 220 days. What is the lifespan of red blood cells in a healthy individual? Let us take a minute or so to collect some answers from the students who are participating, and I will reveal the correct answer at the end of a minute. Okay, I think that's enough time for this question. How long do RBCs, red blood cells, live in a healthy individual? The correct answer to this question is option B, 120 days. RBC's lifespan is for 120 days in a healthy individual. Now, RBCs, where are they produced? RBCs are produced in the bone marrow through a process called erythropoiesis. They circulate in the bloodstream, delivering oxygen to tissues and removing carbon dioxide. So, RBCs are major carriers of oxygen and carbon dioxide. After about 120 days, old RBCs are broken down in the spleen and liver, a process called eryptosis. Eryptosis is nothing but the conjunction of the word erythrocytes, meaning RBCs, and apoptosis, meaning cell death. So, cell death of erythrocytes or RBCs happens in about 120 days inside organs like spleen and liver. Spleen is also known as graveyard of RBCs. Hemoglobin from the RBCs is recycled and iron is reused to form new RBCs. So, hemoglobin is the protein that binds to oxygen and helps in oxygen transport from lungs to the body and from the body back to brain. So, this is a typical structure of a red blood cell, biconcave structure, disc shaped structure with no nucleus, but its sole purpose is to carry oxygen. Now, this is a schematic showing the synthesis or the birth of RBCs inside the bone marrow of a healthy individual. So, in the bone marrow, we have stem cells which then convert into proerythroblast, then goes through stages like early erythroblast, late erythroblast, and normoblast, which is then pumped into escapes the bone marrow. Reticulocytes remain in bone marrow for 3 days, and reticulocyte is then traveling into the arteries, which is the circulation, blood circulation. Reticulocyte will then lose its nucleus to form an erythrocytes. So old and damaged erythrocytes get recycled by the macrophage M5, spleen M5. The macrophages, spleen resident macrophages, eat up the debris and dead RBCs or which fail to develop from the bone marrow. Okay, is it clear to the students that the RBCs has a lifespan of 120 days in the adult body? I hope it's clear to the students. If yes, then let me know in the chat box and we will move forward to the next question. Okay, cool. Moving on to the fourth question. Which of the following is present in RBCs? Again, questions lined up for RBCs. Which of the following is present in RBCs? Option A, Golgi apparatus. Option B, endoplasmic reticulum. Option C, ribosomes. Option D, cytoskeleton. Now, out of which of these cell organelles is present in RBCs? Is it the Golgi apparatus? Is it the endoplasmic reticulum? Is it ribosome or cytoskeleton? Now let's take a minute or so to collect some answers, and then I will reveal what is the correct answer to this question. Okay, it's time for me to reveal what is the correct answer to this question. Which of the following is present in RBCs? The correct answer to this question is option D, cytoskeleton. Cytoskeleton is present in the RBCs, not any other cell organelle. Now let us closely look what is there in the RBCs. In a mature red blood cell, it lacks most of the cell organelles, including Golgi apparatus, endoplasmic reticulum, and ribosomes to maximize space for hemoglobin. So, the sole reason that the cell lacks most of these cell organelles is because it has to accommodate more and more protein molecule, which is hemoglobin, which will enable its sole function of carrying oxygen and carbon dioxide. However, RBCs do have a cytoskeleton which maintains their biconcave shape, provides flexibility, and allows them to squeeze through narrow capillaries. But why do RBCs have a cytoskeleton? Because it maintains its biconcave shape and provides flexibility to the RBC, to this blood cell, allowing them to squeeze through narrow capillaries, blood capillaries. Arteries turn fine into capillaries. Capillaries are the ones where blood and gaseous exchange happen between cells and tissues of most of the body. And what is the component of this cytoskeleton? The spectrin actin cytoskeleton is essential for RBC's durability and function. So this cytoskeleton and this shape of RBC is very important for its function. So the biconcave shape of RBCs increases its surface area to accommodate more and more hemoglobin protein, aiding in oxygen transport. I hope it is clear to the students why RBCs have a cytoskeleton and why cytoskeleton is the correct answer to this question. Okay, very good. Here comes the fifth question. Erythropoietin is produced in dashed. Option A, kidneys. Option B, liver. Option C, pancreas. Option D, bone marrow. Erythropoietin is produced in which of these organs in the body? Is it kidneys? Is it liver? Is it pancreas or bone marrow? You have to recall the facts about erythropoiesis, red blood cell, birth of the red blood cell or synthesis of red blood cells. Where does erythropoietin comes in and which is the organ that secretes erythropoietin? Let's take a minute or so and collect some answers, and then I will reveal what is the correct answer to this question. I think it's enough time for this question, and time for me to reveal what is the correct answer to this question. Erythropoietin is produced in option A, kidneys. Erythropoietin is produced in kidneys, not in the bone marrow, not in the liver or pancreas. Erythropoiesis sure happens in the bone marrow, but erythropoietin is a hormone that is produced by the kidneys. Erythropoietin, short form EPO, is primarily produced in the kidneys, about 90%, and to a lesser extent in liver. In fetal liver, erythropoietin is produced, but in all adult lifestyle, The kidneys are the source of erythropoietin. What is the site of production? Produced by interstitial fibroblast cells in the renal cortex near the peritubular capillaries. So in the kidney, we have the outer layer called the cortex and the inner medulla. In the renal cortex, there are cells called interstitial fibroblast cells which produce this hormone. Liver, in the liver it is produced by hepatocytes and Kupffer cells mainly during fetal development. This is what I was telling you, that in the fetal development it is the liver, while in the adult life it is the kidneys. What is the function of erythropoietin? It stimulates the bone marrow to produce red blood cells in response to low oxygen levels or hypoxia, and helps maintain the oxygen balance in the body. So erythropoietin is kind of a signal which occurs or which is sent by the kidney to the bone marrow to synthesize more red blood cells to balance, to maintain the oxygen balance inside the body in low oxygen condition or in stress. Regulation: low oxygen levels, hypoxia, increased erythropoietin production. Now we know why. When there are high oxygen levels or hyperoxia, there is decreased erythropoietin production. And this hormone is also used widely in clinics to treat anemia or lack of oxygen in the blood in chronic kidney diseases and chemotherapy patients. Athletes sometimes misuse synthetic erythropoietin for blood doping to enhance oxygen delivery. So it is also misused by players and athletes for doping, since it can increase the red blood cell production and thereby increase the capacity of oxygen, capacity of carrying oxygen. So this is a schematic showing that mostly the kidney produces this hormone erythropoietin, and in the fetal development the liver produces this hormone. This is a signal for the red blood cells to make, this name is derived from erythro, which is red blood cells, and poietin means to make. It is signaling the bone marrow to make red blood cells. It matures it when it acts upon the immature red blood cell and turns it into a mature RBC. So this is the job of erythropoietin, and it is secreted by the kidney. Now is it clear to the students? Okay, great. I hope it is, and let's move on to the next question. Question number 6. Platelet cells are also known as thrombocytes. They are derived from dashed. Platelet cells, also known as thrombocytes, are derived from dashed. Option A, megakaryocytes. Option B, monocytes. Option C, myeloblasts. Option D, erythrocytes. This is again related to the blood genesis pathway or synthesis of blood from its precursors. So basically this question asks which cells are the precursor cells of platelet cells or thrombocytes. Are they megakaryocytes? Are they monocytes, myeloblasts, or erythrocytes? This is an informative, easy question. I hope everyone can answer this. Let us take a minute and wait for some answers. Meanwhile, you think about and let me know what do you think is the correct answer to this question. I will reveal the correct answer in a minute or so. Okay, that's enough time for this question. Let us try to see what is the correct answer to this question. Platelet cells, also known as thrombocytes, are derived from option A, megakaryocytes. Megakaryocytes are the precursors of platelet cells, which are known as thrombocytes. Now, let us understand the synthesis of platelets and what is a megakaryocyte. Megakaryocytes are large bone marrow cells responsible for producing platelets or thrombocytes. They are among the largest cells in the bone marrow with a lobulated nucleus. So, this is the megakaryocyte. It has a lobulated nucleus. It means the nucleus is multi-lobed with balls and blobs found in the bone marrow, but fragments of their cytoplasm enter the bloodstream as platelets. So, platelets are nothing but fragments of these megakaryocytes. And the size of these cells are very huge. They are 50 to 100 micromolar in diameter. So let me remind you, the general size of a cell is 1 micrometer, but a megakaryocyte is 50 to 100 micrometer in diameter. So these megakaryocytes have their origin from hematopoietic stem cells, which then turn into a promegakaryocyte, finally transforming into the lobed nuclear megakaryocyte. And such megakaryocytes undergo disintegration of their cytoplasm, which is now entering the bloodstream as blood platelets. So, if you recap the summary of platelet production, hematopoietic stem cells, which are the precursors for all the red blood cells as well as the white blood cells, transform into myeloid stem cells, which give rise to megakaryoblast. And then from the liver and kidneys, another hormone comes into picture called thrombopoietin, which is a brother hormone to erythropoietin. Thrombopoietin signals the production of thrombocytes from the megakaryoblast. So now maturation and endomitosis. Endomitosis means the division of the nucleus within the cell sharing a common cytoplasm terms into the results in the formation of megakaryocytes, multinucleated megakaryocytes with proplatelet extensions. So these are proplatelet extension because these undergo fragmentation, these extensions undergo fragmentation, now forming blood platelets. So this is the whole scenario of a platelet synthesis and development. And the precursor cells for platelet development are known as multinucleated or lobed nuclear megakaryocytes. I hope it is clear to the students what are megakaryocytes and how they lead to the production of platelets in the blood. Great, cool. Moving on to the seventh question of the session. Dashed protein is responsible for conversion of fibrinogen into sticky fibrin that helps bind the clot. I will read the question again. Please look at it carefully. Which is the protein that is responsible for conversion of fibrinogen into sticky fibrin that helps bind the clot? The clot here means a blood clot. Option A, albumin. Option B, spectrin. Option C, ankyrin. Option D, thrombin. Now, which of these four proteins is responsible for conversion of fibrinogen into sticky fibrin in blood clotting? I think this is a pretty easy question. Most of you must know answers to this. Let us take or wait a minute for everyone to come on the same page. Think about what is the correct answer, which is the correct option. Let me know in the chat box and I will reveal the correct answer in a minute or so. I think that's enough time for this question, and it's time for me to reveal what is the correct answer to this question. Dashed protein is responsible for the conversion of fibrinogen into sticky fibrin that helps bind the clot. And the answer to this question is option D, thrombin. Thrombin is a protein secreted by thrombocytes, none other than our platelets, is responsible for blood clotting. Now, what happens in blood clotting? Thrombin factor 2A, which is the other name of the thrombin, is a key enzyme in the blood clotting cascade. It plays a crucial role in converting fibrinogen into fibrin, which forms the structural framework of a blood clot. So, the fibrin forms the meshwork in which blood cells and other dead cells are trapped, forming the blood clot. Now, what is the role of thrombin in blood clotting? First and foremost, the important role is to convert fibrinogen into fibrin. Thrombin cleaves fibrinogen, a soluble plasma protein, into fibrin monomers. These fibrin monomers polymerize to form an insoluble fibrin mesh that stabilizes the clot. So, this is the mesh that I was talking about, which is most important to bind or to form a cement into which cells can be trapped. Thrombin not only does convert fibrinogen into fibrin, it also activates clotting factors like factor 13, which cross links fibrin strands, making the clot stronger, and factor 5 and 8, which amplify the clotting by accelerating thrombin generation. So it's a feedback loop, thrombin generating factor 5 and 8, factor 5 and 8 feeding back to thrombin generation. Thrombin also enhances platelet activation. Thrombin binds to platelet receptors PAR1 and PAR4, stimulating platelet aggregation and further clot growth. So here is a smooth muscle. What is required in the clot? Cytokines, growth factors, autocoids, proteases, shape and permeability change, smooth muscle. Thrombin is affecting everything. Thrombin targets the lymphocyte. Thrombin helps in clot formation, forming the meshwork. It also signals neutrophils. It directly links platelet activation. It activates monocytes and also helps in secreting other clotting factors like cytokines, growth factors, autocoids, and proteases. So, what happens in the clotting overall process? What are the important clotting factors? Whenever there is a damaged blood vessel, injury to vessel lining triggers the release of clotting factors in a broken blood vessel. Clotting factors convert the inactive prothrombin into active thrombin. Now, once thrombin is activated, thrombin now acts as the cleavage enzyme, cleaves fibrinogen, which is a soluble form of the fibrinogen, and turns it into insoluble fibrin. Now, insoluble fibrin polymerizes, many such monomers polymerize at the clot site. So, formation of a platelet plug, vasoconstriction limits blood flow, and platelets form a sticky plug like this once the platelet is activated due to thrombin again. And then formation of an insoluble fibrin meshwork which traps these platelets as well as RBCs. Fibrin strands adhere to the plug to form an insoluble clot. So this is the process of the whole blood clotting, where thrombin plays a major role and which is important for repairing injuries and preventing blood loss. Now, is it clear to the students that thrombin converts fibrinogen into insoluble fibrin during a blood clot? Great. Okay. Now here comes the 8th question. Muscles of alimentary canal and reproductive tract are examples of dashed. We are shifting gears into the muscular system now. Muscles of alimentary canal and reproductive tract are examples of dashed. The alimentary canal or the digestive canal and the reproductive system.
What are such muscles made up of? Option A, skeletal muscles. Option B, visceral muscles. Option C, cardiac muscles. Option D, striated muscles. The muscles of the gut and the reproductive tract fall into which category? This is also a easy direct question, but let's wait for a minute for all the students to come to the same page, and I will reveal the correct answer after a minute or so. Okay, it's time for me to reveal the correct answer. But I see a student has answered option D. Fatima has answered option D. Let us see what is the correct answer to this question. Muscles of alimentary canal and reproductive tract are an example of option B, visceral muscles. Option B is the correct answer. I'm afraid to say that it's not striated muscles, Fatima, but visceral muscles. Now, what are visceral muscles? The alimentary and reproductive tracts primarily contains smooth muscle or involuntary muscles, which are also called visceral muscle, because it is controlling the parts of viscera. Viscera means the internal organs of the body, which is involuntary and unstriated. It can never be striated, facilitating peristalsis, which is the movement of the food through the alimentary canal, and other movements essential for digestion and reproduction. So the correct answer is visceral muscles. Here is the diagram of our digestive system consisting of the alimentary canal, the mouth, upper alimentary canal elements, the mouth, the tongue, pharynx, the epiglottis leading to the esophagus or the alimentary canal. Then comes the stomach, the duodenum, accessory glands like liver, gallbladder, pancreas. Then comes the small intestine, where most of the absorption takes place. Then the large intestine, where wastage is stored, the colon and of the large intestine, and the rectum and the anus. So all of these muscles are the smooth muscles or visceral muscle, which are involuntary, because these are not under our control. Same for reproductive system, where we have the male reproductive organ, the testis, and accessory glands and structures like the penis, the prostate gland, the seminiferous tubules, the vas deferens, and many other structures which are involuntary in nature. The secretions, their function is not under our direct control. So, what are the features of these kind of visceral muscles or smooth muscles? They are non-striated, spindle-shaped, and uninucleated. So, they have single nucleus like this. They are non-striated, there are no markings, and they are spindle-shaped or needle-shaped. Where are they located? Found in the walls of all internal organs like stomach, intestines, blood vessels, uterus, etc. And what do they control? They are controlled mostly by involuntary actions regulated by the autonomic nervous system. So the nervous system, autonomic nervous system, mostly controls the smooth muscle activity. Their contraction is slow, sustained, and rhythmic contractions, and they are regulated by hormones, neurotransmitters like acetylcholine, norepinephrine. They are high fatigue resistance, contracts continuously without tiring quickly, and they have unique gap junctions that allow synchronized contraction of cells. So, such are the characteristics of the smooth muscle which form the viscera or the internal organs of the body. So, there are three main types of muscle: the skeletal muscle, which are striated, tubulars, and multinucleated. They are mostly voluntary muscles. All the limb movement happens to the skeletal muscle, usually attached to the skeleton. Smooth muscle is what was the correct answer, which is lining the viscera or the internal organs. They are non-striated, spindle-shaped, and uninucleated, mostly involuntary in nature and usually covering the wall of internal organs. Then we have a special kind of a muscle called a cardiac muscle, which is only present in the heart. They are also striated, branched, and uninucleated, but they are involuntary, only covering the walls of the heart. Is it clear to the students? You can let me know in the chat box with a yes or a like, and we will move forward. The correct answer to this question was visceral muscle, which is lining the viscera or internal organs of the body, the alimentary canal, and the reproductive tract. Great. Looking at the ninth question of this session, the muscles which have striped morphological appearance can be controlled by nervous system, possess short twitch of contractions, and are present all over the body are. Now this is a dead easy question. Muscles which have striped morphological appearance can be controlled by nervous system, possess short twitch of contractions, and are present all over the body are which muscles? Option A, skeletal muscles. Option B, visceral muscles. Option C, cardiac muscles. Option D, striated muscles. Let us take a minute and collect some answers. I'll reveal the answer very soon in a minute or so. I think it's time for me to reveal what is the correct answer to this question. The muscles which have striped morphological appearance can be controlled by nervous system, possess short twitch of contractions, are present all over the body, are option A, skeletal muscles. Striped morphological appearances, striated muscles can be controlled by the nervous system, yes, and also possess short twitch of contractions and present most in the most organ part of the body. Skeletal muscles are the type of muscle tissue that is attached to the bones and allows for voluntary or conscious movement of the body. They are responsible for a wide range of movements, including walking, running, lifting objects, and even breathing. So, the breathing muscles, the intercostal muscles, are skeletal muscles attached to the rib cage of our body. Skeletal muscles are found throughout the body, attached to bones via tendons. And unlike smooth muscles, which control involuntary actions like digestion, or cardiac muscles, which control the heart, skeletal muscles are under conscious control. They are multinucleated like this, many nucleus, muscle fasciculus made up of muscle fibers, and muscle fiber made up of myofibrils, and each myofibril has this kind of a zigzag or striated patterned appearance. They are multinucleated, they can extend while stretching, they show elasticity, returning to their original structure after extending. They are excited in response to stimuli. They have a contractile nature, meaning they contract when interacting with stimuli. So this is the structure of a muscle fiber, muscle fibril. It has many components like the actin and myosin component. Actomyosin tracts form the basis of muscle contraction. So where does nervous system come into picture? At neuromuscular junctions. At neuromuscular junction activation, an action potential arrives at the neuromuscular junction, motor neuron terminal. Acetylcholine is released, binds to the receptors on the sarcolemma, muscle cell membrane. An action potential arrives at the neuromuscular junction from the motor neurons. ACH is released, binds to receptors and opens sodium ion channels, leading to an action potential in sarcolemma. So when acetylcholine is released from the motor neuron junction, it binds to receptors and opens sodium ion channels, leading to an action potential in the sarcolemma or in the cytoplasm. Sarco is for muscle. Action potential travels along the T tubules, leads to calcium efflux, activating the muscle contraction. The actomyosin complex, thick and thin filament interaction leads to muscle contraction. Muscle shortens and produce tension. So acetylcholine is released, binds to the receptors on the sarcolemma, muscle cell membrane. This opens sodium ion channels, depolarizing the sarcolemma and generating an action potential. Action potential propagation. The action potential travels along the T tubules deep into the muscle fiber. This triggers the sarcoplasmic reticulum to release calcium ions. Calcium release and binding. Calcium binds to troponin, causing a conformational change. Troponin is a component of the actomyosin fiber. This shifts tropomyosin, exposing binding sites on actin filaments. Cross bridge formation and muscle contraction. Myosin heads is another protein, myosin, binds to the actin, which is here on the green, forming cross bridges. ATP is hydrolyzed into ADP plus PI, providing energy for the power stroke. With the utilization of an energy molecule, this attaches and movement of the motor. Myosin pulls actin, shortening the sarcomere and causing muscle contraction. Muscle relaxation. When stimulus stops, calcium is actively pumped back into the sarcoplasmic reticulum, Troponin returns to its original shape, blocking actin binding sites, and the muscle relaxes, returning to its original length. So, this is the whole procedure of a muscle, striated muscle or a skeletal muscle activation from the nervous system through a neuromuscular junction. And we just knew that skeletal muscle is a striated in morphology, is under voluntary control, is activated by nervous system, and undergoes short twitches or contraction using the actomyosin thick and thin filament morphology. Is it clear to the students? We will move forward with the questions then. Very good. Moving forward to the 10th question. Slowly and steadily we are reaching towards the end of this session. The main component of thick filament in skeletal muscle is dash. What is the main component of thick filament in skeletal muscle? Option A, actin. Option B, tropomyosin. Option C, troponin. Option D, myosin. The thick filament in a skeletal muscle, what is the main component in that? Is it actin? Is it tropomyosin? Is it troponin or myosin? Let us take a minute and collect some answers, and I will reveal what is the correct answer at the end of the video. Okay, I think that's enough time for this question. The main component of thick filament in skeletal muscle is option D, myosin. Myosin is a motor protein that forms the thick filament in muscle fibers and plays a crucial role in muscle contraction through its interaction with actin. So, this is the thick and thin filament that I was talking about during the previous question. The thin filament is consisting of actin, and the thick filament here is myosin. The thin filament is actin. Myosin is a motor protein that forms the thick filament in muscle fibers and plays a crucial role in muscle contraction through its interaction with actin. Now, what is the structure of myosin? Myosin is a large fibrous protein with the following key parts. It has a head region or a globular region, contains an ATPase enzyme that hydrolyzes ATP for energy. Since we know that muscle contraction requires energy, so myosin is the protein that hydrolyzes ATP, binds to the actin filaments to form cross bridges. So it will form these cross bridges, hydrolyze this ATP like it is shown here. Myosin heads split ATP, become reoriented and energized. Myosin heads bind to actin, forming cross bridges. Undergoes a power stroke pulling action during contraction. This is the power stroke action. Myosin heads rotate towards the center of the sarcomere, power stroke. As myosin heads bind ATP, the cross bridges detach from the actin. And contraction cycle continues if ATP is available and calcium level in the sarcoplasm is high. Then there is a neck region of the myosin, which is this region, acts as a lever arm transmitting the force from the head to the body or to the fiber. And the tail region is a rod-like region, forms the core of the thick filament, helps in myosin filament assembly. Now the thick filament composition. Thick filaments are primarily composed of bundles of myosin. So these are all different myosin proteins. Each thick filament contains hundreds of myosin molecules arranged in a bipolar structure. The heads project outward for actin binding, while the tails from the filament backbone. Now, what is its role in muscle contraction? Myosin binds to actin, forms a cross bridge, and upon ATP hydrolysis, myosin head pivots, pulling action, pulling actin, the power stroke. Then new ATP binds, so myosin detaches, and the cycle repeats, leading to muscle contraction or sarcomere shortening. So this is how a detailed structure of the myosin thick filament looks like. These are the myosin heads. This is the neck region, and the tail region is embedded in this thick filament. This is the band of the myosin, and over this is the interlinking actin band. This forms a single unit of muscle called the sarcomere. Portion of the thin filament is the actin, troponin, tropomyosin, and portion of the thick filament is the myosin molecule. It has the head, myosin light chains, and the myosin heavy chain, the flexible hinge region, ATP binding sites, and actin binding sites. While in the thin filament, we have actin subunits, tropomyosin binding to actin, and troponin guarding the actin myosin binding sites. Is it clear to the students? If so, then we will move on to the next question. Question number 11. Tropomyosin is a dash-like fibrous molecule. Tropomyosin is a dash-like fibrous molecule. Option A, spherical. Option B, disc-shaped. Option C, rod. Option D, conical. What is the shape of tropomyosin? Is it spherical? Is it disc shaped? Is it rod shaped or conical? Let me wait for a minute to collect some answers, and then I will reveal my correct answer. I think time is up for this question. Tropomyosin is a rod shaped fibrous molecule. Tropomyosin has a rod shape. Tropomyosin is a rod shaped protein that plays a crucial role in muscle contraction by regulating the interaction between actin and myosin. So, where is tropomyosin present? Tropomyosin is present in the actin filament. Structure of the tropomyosin, it is rod shaped, double helical coiled protein. So this is the tropomyosin dimer, double helical coiled, composed of two alpha helical polypeptide chains. You can see clearly the two polypeptide chains, mainly consisting of alpha helices, extends along the length of the actin filament. So it kinds of coats the actin filament. Each tropomyosin molecule spans about 7 actin subunits. Now, what is its function in muscle contraction? In a relaxed state, tropomyosin blocks myosin binding sites on actin, preventing contraction. So, tropomyosin will guard the actin so that myosin cannot bind the actin and cause a contraction. During contraction, calcium binds to troponin, causing a conformational change that shift tropomyosin, exposing myosin binding sites on actin. So troponin tropomyosin forms the actin guarding complex, which is disrupted via calcium binding during a action potential firing at the contraction. Myosin binds to actin, leading to muscle contraction through the sliding filament mechanism. We just studied the contraction mechanism of the myosin in the previous question. So for an enlarged image, this is the actin thin filament, and the red band here, the double helical coiled protein, is the tropomyosin, along with the complex of troponin guards the actin from myosin binding sites. Now whenever there is enough calcium which is triggered as a result of action potential, calcium binds this troponin, disrupting troponin tropomyosin complex, thereby enabling myosin to bind to actin and perform the contraction. I hope it's clear to the students. If yes, then let's move on to the next question. Question number 12. Dash muscle contractions can be observed in bicep muscles. The bicep arm muscles. What kind of muscle contractions can be observed in bicep muscles? Option A, twitch. Option B, tetanic. Option C, concentric. Option D, eccentric. What kind of muscle movement, what kind of contractions can be observed in bicep muscles? Okay, time for me to reveal the correct answer to this question. Dash muscle contractions can be observed in bicep muscles. The correct answer is option C, concentric muscle contractions. Now here is the summary of all kinds of muscle movements. Muscle contractions are concentric contraction, muscle elongation are eccentric contractions, and then muscle contracts isometric contraction. So a concentric contraction occurs when a muscle shortens while generating force. In this case of biceps brachii, this happens when you lift a weight during a bicep curl. So when you lift a weight using a curl of the bicep, then you perform a muscle contraction which is a concentric contraction or shortening of the bicep muscle. When it is elongation, it is an eccentric contraction, while in an isometric contraction, when there is no movement but you are still holding a weight in your arm, you have an isometric contraction. Shortening of the bicep, elongation of the bicep, and stabilization of the bicep or no movement of the bicep are all these three types of contraction: concentric contraction, eccentric contraction, and isometric contraction. Is it clear to the students? If yes, we will move on to the next question quickly. Question number 13. The dihydropyridine receptors which functions in the skeletal muscle is a dashed. The dihydropyridine receptor, the DHP receptors, DHPR, which function in skeletal muscle is a dashed. Option A, calcium dependent voltage channel. Option B, sodium ion dependent voltage channel. Option C, potassium dependent voltage channel. Option D, chlorine dependent voltage channel. Which kind of channel is a dihydropyridine receptors which is functioning in a skeletal muscle? Let us take a minute and try to answer this question. I will reveal the correct answer in a minute or so. Okay, it's time to reveal the correct answer to this question. The dihydropyridine receptors which function in the skeletal muscle is a calcium dependent voltage channel. Now, what is this DHPR receptor and where is it functioning? In skeletal muscle, the dihydropyridine receptor or DHPR is a voltage gated calcium channel. Acts as a voltage sensor triggering calcium release from the sarcoplasmic reticulum. So if you remember the whole procedure of a muscle contraction, due to action potential at the neuromuscular junction, there is release of calcium from the sarcoplasmic reticulum. So this receptor is involved in the release of calcium from this sarcoplasmic reticulum for excitation contraction coupling. So this is the voltage gated calcium channel, acts as a voltage sensor triggering calcium release from the reticulum. The DHPR is crucial for the process of excitation contraction coupling in skeletal muscle where a nerve signal triggers muscle contraction. The DHPR functions as a voltage sensor, meaning it responds to changes in the electrical potential across muscle cell membrane or the sarcolemma. When the sarcolemma depolarizes, becomes more positive, the DHPR undergoes a conformational change which in turn triggers release of the calcium ions from the sarcoplasmic reticulum. Now, what interacts with the DHPR is a ryanodine receptor, RYR. The DHPR interacts physically with the ryanodine receptor, a calcium release channel located on the sarcoplasmic reticulum membrane. This interaction is essential for the calcium release process. Like this, depolarization induced calcium released, dihydropyridine receptor, this is the receptor, this is the membrane, there is voltage which is sensed by this receptor, which is physically connected to the RYR receptor, ryanodine receptor, which is a calcium channel leading efflux of calcium from the sarcoplasmic reticulum. Now calcium induced calcium released, voltage gated calcium channel is not functioning much, also act as a calcium channel. It drives influx of calcium, increases the calcium inside and also from the sarcoplasmic reticulum. So again, winding up the whole muscle action potential, which is propagated into the T tubes. Here it connects to the sarcoplasmic reticulum with the DHP receptor and the ryanodine receptor, which is leading to efflux of calcium from the sarcoplasmic reticulum. This calcium binds to the troponin tropomyosin complex, exposes actin to bind to myosin heads and results in muscle contraction. While in relaxation, calcium is pumped back using an ATP inside the sarcoplasmic reticulum, restoring troponin tropomyosin complex at the actin and thereby releasing the connection of myosin to the thin filament. Now, is it clear to the students that DHPR is a voltage gated calcium channel which is sensing the voltage of the action potential and leading to calcium release from the sarcoplasmic reticulum? If yes, then let's move on to the 14th question, the second last question of this session. After this is the 15th question, which is the last question of this session. Dash movement from the muscle generate an action potential that leads to depolarization of the inner muscle fiber. What kind of ion movement from the muscle cell generates an action potential that leads to depolarization of the muscle fiber? Option A, sodium calcium. Option B, sodium potassium. Option C, sodium chlorine. Option D, calcium potassium. Which of these ions move inside or outside the muscle cell responsible for an action potential generation leading to depolarization of the inner muscle fiber? Is it sodium calcium, sodium potassium, sodium chlorine or calcium potassium? Let us wait for a minute and then revisit the question. I will reveal the correct answer in a minute. Okay, I will reveal the correct answer to this question. The movement of sodium potassium ions from the muscle cell generates an action potential that leads to depolarization of the inner muscle fiber. It's very easy to remember. Sodium potassium ions are always linked to depolarization in an action potential. Now, what happens during muscle contraction? An action potential is generated when there is shift in ion movement across the muscle cell membrane. For depolarization, sodium enters the muscle cell, making the inside more positive. This triggers the action potential. And for repolarization, potassium exits the muscle cell, restoring the negative charge inside. This Na plus K plus exchange is essential for muscle fiber activation and contraction. So here is a action potential
of the muscle cell, where sodium channel opens, the the voltage becomes more positive, drives it positive more than the threshold, crossing even 0 to positive 30 millivolts. Where sodium channel close, there is a sharp decrease in the voltage. This peak is called the action potential. When sodium channel close, potassium channels open. Potassium channels opening lead to efflux of potassium ions and restoring the voltage back to minus 70 millivolts. So this is a whole skeletal muscle action potential occurring due to sodium potassium ions. So now again revisiting the key events in muscle activation and contraction. First, there is a neurotransmitter release of acetylcholine at the nerve ending in the synaptic cleft. Acetylcholine binds to acetylcholine receptors on the muscle cell membrane, leading to depolarization of the muscle cell membrane or the sarcolemma, because these acetylcholine receptors trigger the opening of sodium channels. This triggers an action potential that travels along the T tubules. So these are known as T tubules, specific to the sarcomeres or the muscle cells. Now in the external surface of the T tubules are these DHPR receptors. The action potential activates L-type calcium 2 channels. These channels stimulate the RYR receptors on the sarcoplasmic reticulum. So these are nothing but the DHPR receptors. Calcium is released in the cytoplasm of the muscle cell. Calcium binds to the troponin, tropomyosin complex, exposing binding sites on actin, and myosin heads bind to the actin, forming actomyosin cross bridges. This leads to muscle contraction. Now in relaxation phase, calcium is pumped back from the cytoplasm into the sarcoplasmic reticulum via SERCA pumps, SERCA pumps. This promotes relaxation of the muscle. So this is a whole summary of the muscle contraction and the action potential that is generated due to sodium and potassium ions. I hope this is clear to the students. If so, then let's move on to the last question of this session. Question number 15. Myosin is a hexameric protein which consists of dash. Option A, 3 heavy chains and 3 light chains. Option B, 4 heavy chains, 2 light chains. Option C, 2 heavy chains, 4 light chains. Option D, 2 heavy chains, 2 light chains, 2 main globular chains. What is the structure of myosin, which is a hexameric protein? What number of heavy and light chain do myosin possess? Let us take a quick minute, since we are coming to the time limit of our session, and try to answer this question. I will come back in a minute to reveal what is the correct answer to this question. The time is up, and let me quickly reveal what is the correct answer to this question. Myosin is a hexameric protein which consists of 2 heavy chains and 4 light chains. This is the correct composition of the myosin hexamer. Myosin is a hexameric protein, meaning it's composed of 6 subunits, 2 myosin heavy chains, as we see in this diagram. These 2 coiled domains are from the heavy chains, which are called MHCs or myosin heavy chains, and 2 pairs of myosin light chains. So we have 2 different light chains and 2 pairs of it. The heavy chains are the large motor containing subunits responsible for ATP hydrolysis and actin binding activities. So the heavy chain has these head molecules which are responsible for ATP hydrolysis as well as actin binding, and a long tail domain. Light chains are smaller subunits that regulate activity of the myosin motor, help in the movement of the motor. 2 types of light chains. One is a regulatory light chain, which is this blue in color. These are involved in regulating myosin motor activity, often through phosphorylation.
and essential light chains. These are involved in structural integrity of the myosin molecule and may also play a role in interaction with actin. So these are the function of different monomers of the myosin molecule. It is made up of 2 heavy chains and 2 pairs or 4 number of light chain molecules. And the function of these protein, this myosin hexamer, myosin is a key protein involved in muscle contraction where it interacts with actin filaments to generate force. And in non-muscle cell, myosin also play a role in various cellular processes like non-muscle cells such as cell motility, cytokinesis and intracellular transport. So these are all cargo transporting motors, cargo motors working on actin cytoskeleton on microtubules, kinesin, dynein, myosin, these are all motors. They also function in non-muscle cell and in muscle cell play a major role in muscle contraction. I hope it is clear to the students. Okay, very good. With this, we come to the end of this TA session. Practice problem set week 9, sorry. I will stop my presentation and turn on my video. So with this, we have come to the end of the practice problem session. Thank you for joining, and I hope it was a fruitful session. We got some concepts clearer, we solved some questions, and hope to see you in the next practice problem session of week 10 next Saturday 3 to 5 p.m. Till then, read your lectures and follow these slides. This is a recorded class and the recordings will be available on YouTube. The link will be shared to you, while these presentations will be uploaded in a Google Drive and the link will be shared to you by the NPTEL authorities. Thank you for joining. I will end this class now.